Okay, I'm updating and you guys are updating the package. So I'm going to lay out the outline of today, what we are going to do. We will first to set up this toolbox environment to run R, okay? So that you can run R script from MATLAB or from the toolbox itself. We have an interface. So a lot of function has been developed in R. You don't want to reinvent the wheel, but of course, I like you to re-implement from R to MATLAB, and you learn. I do this all the time, but sometimes you just don't have time, and you want to call R package or script to get the output, and we can put it back to MATLAB environment and to keep running using it from our environment. So I will show you that. Then next is we will go through the lecture information, which when you install which step?
Just copying and pasting the installation into MATLAB.
Oh, the last step. I mean, that's the save this. When you have this save the parse, if you don't have an admin right, you will get in trouble. Not trouble, but just not be able to save. But you can use this the last. Instead of using save parse, you use that one. Yeah, we'll do the same. So the question from an on-site student is we cannot save parse because we don't have administration privilege. Okay, in that case, you have to use this long command line to save the user path in the local file here. Okay, so next time you still have access to this. Okay, so we will go through lecture information that's for the decomposition. Okay. Decomposition. My third, okay. 3, we will have ICA example code. ICA independent component analysis to separate mixed signal into its original source. The last, we will look at spatial transcriptome atomic analysis. It's a preview of a tool I developed of a single cell gene expression analysis toolbox _ST. Okay, so this is an extension of the current toolbox. So gave you some additional function you can use for spatial analysis. It's still under development, but I want to show you some early stage of the development. Okay, any questions, comments here? Okay. Now, if you have this installed, let's try. Two, you should have this. Example file, okay? Good. You can either use process, raw data, it doesn't matter. Okay, so you see from here, you may want to use some external R package to process this. And here we have a menu called external. And first you want to check your R environment, whether R has been installed in the same machine here. So I'm going to try if I have R installed. Okay, looks like I do have R installed before, but this is a little bit old package. But this should be okay. But I'm not so sure on your computer whether you have this, but I just assume there's no R executable can be found on your local machine or on your machine in the computer lab. So what do you have to do if you are in the computer lab here? You have to go to Zen Network and to find RStudio and R. Okay, so this package, it's a bundle package, has been combined from R language and also RStudio. So I'm going to run it anyway. So I have an older version of 3, but this should be allow me to upgrade to version 4 or higher. So I'm doing this anyway. So then you can see the program here should be able to detect the newly installed R environment. So once you have that, some kind of R environment or executable file has been installed, then you are ready to call those functions with require R under the same menu. Is there any questions from online students? Okay, so the pop-up window tells me, and I have already installed R and RStudio on this machine. So now I will come back to this page and see, check my R environment. Now I have already successfully upgraded my current R version into 4.13. I believe this is the latest version in R. Okay, the old one has been overwritten. Now I can see, I want to run a program to so-called Surrat SC transform. Okay, you can see like this. So this is a normalization method. You remember we have a library size normalization, but this is another more advanced and the normalization method, and based on the modeling of the data. Remember the modeling of data, we want to figure out every gene, how many zeros in there, fit the parameter may be shared across genes. But anyway, this method has been proposed and become very popular. So we want to use this to, rather than to do the simple library size normalization, we can say, okay, we want to do a SC transform. Then we can select this. The first message you will have when you run external function is, are you going to run it to normalize expression matrix? You say yes, then it will go through. If you say, I want to take a look what's going on, you can view the script. That's the R source code I'm going to use. Okay, so this R source code, if you are R person, you should be able to read it. So that's call those libraries and their communications between MATLAB data and write into R. So I write the input data, which input data is this matrix here. And in R, we will read that into R, a variable, then to do this transformation in R. And then the output will be write out. Okay, so if you think this is safe enough, and we can just click OK and to execute these files. Oh, I got it. It must be a typo. So you can help me to fix this, and we can come back again. Yeah, so yeah, so the problem now is we don't have Surat package installed. Okay, that's why this program stopped here. Okay, what you can do is to go to the RStudio here. And then we will install the package. So how to do it? Install. So we have to go to the external R environment to install Surat package. So we can go back to the MATLAB environment. So to call that script. Okay, on my machine are Surit already installed. So I'm going to come back to this to check if I can run this. I probably also need the library of those, the matrix and R MATLAB, but I will try. If I still have error message, I will go back to the R environment to install those two packages. So you can somehow look at the background. There are output comments that's from R is showing what's going on there. Okay, here I'm already done. So you have two options. Either you want to use transformed X, which to replace where the original data which show on this window, or you may want to just export X as a variable and you save it in the workspace. In this case, I'm going to update. So the X matrix in this figure will be updated into its transformed version. Okay, it's done. So now my data has been normalized or transformed using R package, Surit SC transform. So I can do downstream analysis, including t-SNE or UMAP, whatever method you think should be as a follow-up. Okay. Or you can run the whole workflow. So Surit can do the clustering, can do the embedding. So if you decide to do the whole workflow, the transformation are already there. You can look at the source code, and then here, after the transformation, we will run t-SNE, UMAP, found neighbors, and found clusters. So this is a whole solution you just borrowed from our package. So I can run this and see what you can get. So you can take a look of this comment window, and you can see all this are the feedback from the messages from R, and at which step we are now in. And I am doing this transformation again. I shouldn't do this because already transformed, but it's just as an example, and it's okay. Okay, the whole workflow, the process has been done. And you can see you have used, basically you have used R serial package to identify embedding the cells and also identify those clusters. Because you have done both t-SNE and UMAP, you can look at this multi embedding view. So this is a t-SNE versus UMAP for the same population of cells. Okay, that showed you before. You can highlight some of the cells and look at their corresponding position in the from a UMAP. You can look at where the cells are embedded in t-SNE or vice versa. Okay. So that's the idea of using R. And here you can also look at whether you have Python installed. So in my case, I don't have a Python environment has been detected on my machine here. So it's empty, but there's a way if you install Python, then those external comments or packages or functions can originally developed in Python should be able to use here. Okay, we are done with this multi-language integration. Basically, you transform your input data from one language to another processes and get the output from another language and continue to process using the MATLAB environment. And this menu can be expanded, depends on the requirement. If you have some killing program you really want to use, and here, let me know, I may incorporate them into this menu here. So those are just selected. Like Monaco people want to use to do pseudo time analysis and very popular, so I included them here. Okay. Any questions from online students?
I got an error when I tried to do it. It said that there was an error while evaluating menu callback.
Yeah, can you post your, I don't know how to fix that. Can you post your message onto the chat so I know
Okay.
what's the, which comment that you used?
It was, okay.
Transformation?
Yeah, it was in GUI callback run Siret. Sorry, I have it on my laptop, so it's going to take me a while to copy this over. But
Yeah. So probably there are some communication issues between MATLAB and the R you installed. Well, it's very difficult for me to do this diagnosis remotely. I have to look at your computer to see what's going on. Yeah, so so you can, for this one, it's a typo in this function where you can click this line 15 and correct this typo into a return. So I supposed to write as return, but I didn't make it correct. Yeah. So I show you. So this is a line 15 of this function. So if you do go back, so here I come to the function and found this line 15. I already correct here on my machine. It's a typo. It's supposed to be written, but I write is write run. And of course, it's so this problem has already been fixed. I just upload a new version in online. So if you reinstall, you should have correct version of this.
Okay.
But in general, this tells you there's error message which you have a problem to run this. You have to fix the problem anyway. Maybe it's installation problem. You stop you run this successfully. Okay, we have to move on and to the next topic. Okay, we are done with the setup of the environment. So we are going to look at matrix decomposition now. Good. Now I'm going to refer back to the lecture notes and this to identify those decomposition examples I mentioned during the class. I think we should first give a detailed look of this NNMF function. Let me shut down my R environment. Close the network. So let's see a documentation of NNMF. Let's see which, and this is a function inside the toolbox of statistics. Statistics toolbox, and do doc. The function was put under the topic of dimensionality reduction and feature extraction. the general description of this and how the loss function is defined. Okay, so that's a D. You want to minimize this. You can copy the source code here. So just load example data, fisheries. Let me clean my workspace and then load. So that's my input data, and it's the variable called miss, and miss has a 150 rows and 4 columns. And then in the source code here, they recreate, you try to reproduce the same results on different machines. So you put a random number seed reset to 1, then run this here. 2 means how many components you want to learn from the data. Okay, so if you run this, you get the same answer, W and H. You can get from the original. So W and H are so the size of W is 150 by 2, and H is 2 by 4. So the product of these two will be 150 by 4. So I'm trying to do to look at the matrix. So that's the value for this matrix, scaled, and also show in this different color combinations scaled. So this is the values of our original matrix. You can see here. Okay, you have 5, which is higher, and then gradually for the first variable, it's become greater and greater. The second variable start from a 3.5, this color, and the third variable start from 1.4, and there are several blocks in here, and the second variable looks like it is purely random. There's no structure in there. And the fourth variable starts from 0.4 and become higher and higher. Yeah, so that's the global view of the input matrix. Now we are going to look at the reconstructing. How can we reconstruct the original matrix? We have W and H. I want to have an answer from online students. Okay, so the object for an MF is try to minimize the difference between these two, your original matrix and it's decomposed product. So in this case, we have a small value because they are very similar. So to conform this visually, we want to use the same imaging as C function to look at the product of these two, right? So now I can, what I can do is to put this product here and then make a new figure. Okay, so this number 2 figure is what I reconstructed. Okay, so I put the original in the left and reconstructed at the right. So you can see we already using just 2 components to capture the most structure nicely, and almost indistinguishable from these 2 matrices visual representations. All right, so I'm going to close this and to use subplot to do the whole thing. I'm going to use subplot and 1 row, 2 columns. The first, I will have image SC needs. Okay, so this will create a subplot and put in the first position here. And then I will do subplot number 2, W and H. Now you have this left and the right to color scaled matrix representation on your left and right panels here. Okay, we will move on. So this is how are you going to use MF. And the Python version here. Okay, so if you in the early stage, if you have this matrix observed, then you can immediately do this. Just use 1 line of code. You should be able to add your interpretation. You probably have a very nice paper at that time. Review. Okay, Eigen Decomposition. So I will just use this very simple examples to do eigen decomposition. So my A is 1, 2, 2, 4. Okay. So let's look at the function. So you have A, then you have a eigenvector and eigen value as put in the V and D. So you have V is the second. So this is eigenvector, and this 5 is eigenvalue, and 0 is not an interesting one. So we will just keep that. Let's see. So we have a relationship. That's A times now we have this relationship A times this V is this D. Okay. So A should be able to be reconstructed with V D times V. So we just try if we can do this here. So I know what my A is. So I'm going to the inverse of V times D times V. And then this should be able to reconstruct my original A. So that's how you decompose A into its eigenvalue and eigenvector, the product. Again, A have to be squared matrix. If you have random matrix, say 3 by 2, and if you do eigen, you get error message. Okay. If you do this, it should be the same. Okay, the same as we have this equation here. So any problem, any questions regarding eigen decomposition? Okay, I just use a very simple example, show you the numerical validation. Okay, it is the case. Oh, good. Thank you, Alessa, for the feedback. SVD. So SVD is a big brother of all of the decomposition method. It's flexible, versatile, and can handle any kind of matrix. I believe I already showed you on in lecture. This is the case if you do SVD for the matrix. Okay, now let me clean up my desktop here. This time A is 1, 2, 3, 4, 5, 6. Okay, non-square matrix. And we decompose this into a U, S, and a V. Okay, SVD. Three matrices. U times S times V transpose is A. Okay, we decompose it and multiplicate them and together to get the product, which is reconstruction of original matrix. Let's look at what's inside the U. U is a 2 by 2. The reason is your original matrix is 2 by 3, so this is a 2 by 2 matrix. If you do A times A, so this will be let me consider this will be a 2 by 2 matrix. Is this 2 by 2? So here I'm going to do an eigen, so that's small u, small d. We don't need a d, we just, oh, okay, so let's see. Yeah, so I will repeat what I did. So I get the SVD and then look at U. Is this, if I do small u, rather than to do a eigen decomposition for original matrix, because original matrix is not a squared matrix, but I do this convert, make it into a covariance matrix. This is 2 by 2, and then I can do the eigen decomposition. You will get the similar answer. So you can see numerically there's the same. It's just direction is slightly different. And that's why you can use this method to compute U, or in other way around, you can compute V. Okay, in this case, you do this. So this V will be similar to the V you get from SVD. Okay, can you see the similarity between them? Numerically the same, it's only the direction, the sign is different. Okay, it's time for a break, 5 minutes, and we will come back to see the low rank approximation using SVD's results. Okay, let's come back to look at this low rank approximation. We certainly can use this original, this is simple example, only have 6 values in there, this 2 by 3 matrix as an example. So you can use first component, use the first and second component to approximate it. But here I want to use a more complex input matrix to do this. So let's just clean up the environment here. I will load this fishery data again. So I'm going to do the SVD decomposition for this A matrix. Okay, instead of A, our input is Ms. Okay. Let's look at the S matrix inside. That's in the middle. You see it's already ordered by its singular value. Basically, it's a scaling factor. So to that direction, how to rescale the data to fit the original data. So you can see the first component takes 95.95 value, and then second one reduced to 17, and then the last one only have 1.8. So we probably can be very easy, very safely to remove the last component, or maybe last 2 components. So let's see first, if we don't remove anything, and then we can reconstruct the original matrix. Okay, just like what we did, we can see figure the first, that's original, right? And then we will have subplot. This time, instead of using W and H, we use U times S times V transpose. Okay, achieve this similar reconstruction, and highly similar to its original. Okay. We can also check, use this norm here, instead of using the decomposition MF, here we reconstruct our matrix. Use this, is very close and almost 0. So okay, remember it was much larger than this. So this is very close approximation, basically. The difference almost 0. Okay. Now let's use only the first component to reconstruct. So I'm going to use only the first row and only the first value. The first column. I think this one should be, let me look at first column. So in this case, I only use the first component. Okay, this is my reconstructed. And the difference between hope you can, yes, let's just, yeah. So on your left side is still original matrix, but this is reconstructed, but just use one component. The difference, if you use only the first component, then the first value here was first component. So the difference, this should be larger than you use the full spectrum. So here is 18, okay? The difference between these two matrices. Now I can use first and second. Two components to reconstruct. And the difference between my original and this reconstruct should be smaller. So the value should be much smaller than this. Okay, 3.9. And the figure, subplot number 2, if I use two components, okay, so now it looks like this. It's already close to the original, but you see the difference between this section and that section is not so obvious, and this part is not. So, but if I use two components, and then you see it's much clearer separation between this subdomain or subclusters of these variables. Okay, you can keep changing and see I can do top 3. Adding more components, add more components in the norm. First 3. Okay, this becomes smaller and smaller difference. Okay, so that's a low rank approximation. So you can use different numbers of the truncated components from the U, V, and S to reconstruct your original data set. Okay, so next we are going to move to ICA. Here I need you to download example data, example code. I already put that code into the Google Drive. So let me copy the Google Drive folder into chat. Okay, either this one or that one. I select one. The file we need is called PCA ICA zip. So if you select this and download it. Okay, here I download this file. And now it's located in my downloads folder. Don't need the rest, so I delete them. I right click and extract them into a folder. Okay, this folder contains some of the demonstration code. So this is a example demonstration. I want you to take a look of this demo ICA. And MATLAB suggests to open this as a live editor, live script. I accept it. So now the original script has been annotated and separate into different sections. And click, see if I can run it. Yeah, so I really need to change my folder into this. So here I copy the folder name and also CD into it. So I change my folder to this place, so I can have all access to all the function under this folder. So the complaint of this running error is fast ICA function cannot be found. So I switch my current working folder to here. I can copy this folder, but you have to change this user to your name. Okay, depends on where your download folder is. But anyway. Okay, I come back to this page right now, and I can run this section by click run section. Okay, it show up this IDC decomposition. That's true signal. You have 3 different sources and the mixture and the ICA reconstructed. Okay, the PCA does not be able to recover the original signals from observed mixture. This is a synthesized example. You know what's the shape of the input of these three sources, and then you have some noise to be added into this, and then we reconstruct. So reconstruct by using this function and add noise. And then to reconstruct through this function, fast ICA, and give you the output. And they tried other methods, PCA, and the plot results into this panel, 5 panels. Okay. So the next one is an example of audio information. Then let's see whether we can run this. Okay, I have one source, 2 three different sources, and the mixture number one. Okay, mix number two. Mix number three. And then we use ICA to decompose and reconstruct it. Reconstruct number two. Reconstruct number three. Okay, so you can actually do it. Then use different method. Also works. Well, so this ICA method, this algorithm doesn't work as clean as this fast ICA algorithm. Yeah. So that's you have to look at the source code in order to understand all of this. But yeah, this example I found is very interesting, and play with it. I want to know whether you can run it. No, I don't know. Here, maybe we have another example. Yeah, I don't know what they are trying to do here. Anyway, so this is another example. We try to show how to get this one. Pre-mixed audio. Okay, I just gave you some time to look at these examples. Then we are almost there to move on. Okay, we are done with this section. And then right now here, it's almost done. Let's just spend another 5 minutes to finish off this ICA example. By the way, anyone have any questions about your homework project?
Yeah, you wanted us to kind of more or less use the lasso command, I think, right? You said it was more open-ended, but
Yeah, it's open-ended. But if you want to use lasso, so what kind of question you want? Lasso is a regression method. So what you want to regress with, and what's your prediction, and the response variables. So you can define any questions you want. As long as it makes sense, and you can explain the meaning of your analysis.
so were we supposed to create a model, or were we just supposed to more sort of identify the genes of interest? I guess they're kind of the same, but
Yeah, yes. Also, you want to identify those genes which contribute to your classification power, and then look at those genes and then see whether expression program associated with the genes, what their function are. That will be a good direction. I mean, this one of the directions. And this, yeah, it's supposed to be everybody has your own solutions. If you want to have a teamwork, then you have to tell me which part is your teammate contribution, which part is your own. It's not. The final project is a presentation. And you can, so this is not a final project. It's just small homework. Yeah, this is a homework project. For the final presentation, normally I ask students to present a hypothetical study and say, okay, if I had this data set, I want to do what kind of analysis, or you want to propose a study and using the method we discussed through the course. Yeah, so either it's a model project or it's a proposal, what you want to do if you have that data. A 10 minutes presentation with 5 minutes for questions. That's the final presentation we will do in the early May. Okay, now we are going to look at some of this prototype of single cell gene expression analysis tool for spatial transcriptome. So I'm going to send you a link so you can so that's what I want you to do. So let me clean up here. So found a folder. So this is a good place, download folder. And then unzip this to your folder. So I copy this to the chat. So that's in MATLAB. Okay, what I'm doing here is I found a folder which I can download this package and put here is temporarily. I just unzip this function. Okay, once this is done, you have the folder called the Spatial Transcriptome main. Just double click and inside here, that's all we need to do. Again, this is an incomplete project, so don't expect you have very comprehensive experience. I just want to show you what we how we think about this data and what the structure looks like and what we can do with it. Okay, if you are here, there is a test. Okay, that's what we can try. Let me know if you need more time. I can wait for another couple minutes. Here are just a couple of example data, and we can download directly from a geo database and using this accession number to read gene expression data and also the image, and then visualize here. I don't know if that will work right now here. Oh, I needed to have a, yeah, so let's just try. I run this section. Oh, something wrong. Okay, the first example doesn't work, so I'm going to try the second one. Okay, do you have this? Let me know anyone have got the same. So on this window, you'll see this tissue section image. And also that's the transcriptome data points. Okay, each pixel here is a probe sampled by this probe and touch on this particular tissue. Before you sample them, this tissue has been, the picture has been taken and to give that picture, and every pixel has a corresponding position on this. Okay, you're supposed to be able to rotate this. Okay, now the color indicate the library size. So how many rays you sampled from this particular probe. So this position looks like it's more active and a lot of genes has been expressed in a high level. So you get more rays in this particular yellow region. Okay, we need to somehow align this probe image with the tissue section. So now you see it's not well aligned. We need some operation to zoom in, zoom out, and flip the image. So they can, for example, this section here should be this part, right? So I needed to do a rotation So here are some buttons for you to do the rotation, for example, left, right, down, flip. Yeah, so this button here is the rotation. So I'm going to click it and to rotate. Yeah, so I try to rotate the tissue sector, but looks like it's not enough. I probably have to do the flip because I rotate, but it hasn't been able to align very well. Maybe this okay. I don't know how to. So okay, this section should be here, right? So I'm going to rotate an image again. Not yet. Yeah, I'm pretty sure we need to do a flip. To flip this so you can match this section with that and this block here. Maybe here. So I'm going to do a flip and see whether I can get it correct. I'm going to flip by x-axis and then rotate again. Line 8 on which file number 8? Alex?
Oh, it was this code test.m. Yeah, it was just that line 8.
Yeah, so I don't know your specific error message. I need to know. But it seems haven't get this worked. So if not work, you maybe try this example.
Okay.
You can uncomment this and using this as input, see whether you can get anything. But here I have some problem with alignment between my sample and I don't know how to flip. How to rotate. So I make it smaller. So okay. So Alex, are you in? Okay, let's error message. Undefined function for input char. Alex, are you in this folder here?
Yeah, I have it opened. I don't know if there's a special command to make it an active folder.
No, you
something?
you just need to unzip this, and also you have to be in this folder as a working folder. So you have to be here. So you have access. You can look at this function here. Yeah, I want to play with this second example because this may be easier to align. I have some difficulty to align this section with my example. So so that's the other message I think you get. Okay, let's see.
Yeah, I just had to double click on it. Okay.
Yeah, sometimes it's just connection issue network. So, so I got the second image here. So this looks like easier to align. I don't have to do anything. It's already aligned basically. But I wanted to make it smaller to make it comparable. And I also need to move up, down. So anyway, at the end, you should have a picture which covered your section with those spots and aligned. Okay. So that's the first step. Okay, so right now it's the package is not yet there, and so by it's good enough to just show the idea. So we have this aligned structure here. Then you can somehow do a 2D, 3D, or if you don't need that, you only look at this. So some of these function. Yeah, so Alex, I believe you have a problem because you don't have this function installed. If you don't have this package, so if I do which this function is, so you need a toolbox called image processing toolbox.
Okay.
So this image rotation, this function is inside that toolbox. Alex, I believe on your machine, you don't have this
Right.
image processing toolbox installed. I think that's a problem. Now what we can, we look at this, and after you have done this basic alignment, yes, Alex, you have the same problem. And what you can try is to do this add-ons and to find image processing toolbox and see whether you have the right to install it. So I show you where it is. So in this search, if you do the image, you need image processing toolbox to be installed.
Okay.
Along MATLAB. Remember, we need statistics and machine learning toolbox. But in order to do this spatial and the processing of the image, you need this toolbox. In the computer lab here, all those toolboxes have been installed. So that's why I can have this rotation options. But anyway, so those just principle, I just introduce a principle. So you have this structure aligned, so you know where it is, the corresponding position in the slides. And then we have a couple applications. First, you want to show gene expression level. So right now, the spots indicate the library size. Okay, that's the global expression, sum of expression. Here you can click this green box button to select a gene. And if this gene is highly expressed, it's ordered on the top here, and I will find a gene which express a little bit. So that's the expression level for that gene. So you see this gene highly expressed in this particular spot. And then you may want to see, okay, what it is, where it's located. So this gene highly expressed in those locations. You can try other genes. That's the expression level for that particular gene. So some genes expression may be more interesting than other, maybe follow the structure of those tissue sections. We want to identify those genes if you can. There are certainly algorithms can help you to find those genes. But at least you have this capacity to look at gene expression. For this example is expression of that particular gene. On this picture, you can do the same to look at the gene. POE. So you see its expression distribution. I think this is one of the most important feature we need to have in order to manually check the gene expression distribution for individual genes, because every spot here has features of all these genes. So you want to know where the expression of this. Okay. So that's one, and then next one is to look at the marker genes for brushed cell. Okay, let me use this as an example. For example, I'm interested in a particular section of the the tissue. I'm interested in, for example, this part of the tissue and see what expression level is high, which gene expression level is particular high in this particular region. I can highlight this and then say, okay, tell me which gene, give me top 10. Let's compute, okay, you found highly expressed gene for that selected part. Okay, this is not so obvious because maybe this particular tissue is not so sected, and we found some of this gene, for example, this one, and highly expressed in this spot. Okay. All right, so that's one of the example. You can look at the function of that gene. That's another example. Okay, third example. By the way, I used the lasso to do this analysis to identify the gene. So those are the example marker genes for a particular region selected for. So next, again, you can take a video for this and also this call. So here you will come to no. Yes, I have to shut down. So this function shouldn't work. Okay, so this is an incompleted functions. Don't touch it. And this one we have some other functions to do this image processing. And I want to borrow some image processing built-in function to do the clustering automatically. So this part is try to more sensitively, systemically identify different sections, and rather than to select by hand, you can define different clusters. And then for those clusters, we can look at marker genes rather than you select manually which section. In this case, it's very difficult for you to select all these endothelium cell layers, for example. Okay, that's all I think for the introduction of this. So again, this is not, it's still a project ongoing, so not ready for the real world application, but just let you know there's something here for you to later maybe you can use. Okay, that's all for today, and let me know you have any other questions. See you next week.